Hello and welcome to Sam online class. This is Dr. Subrat Kumar Mahanti. Friends, today we shall discuss about transcription and translation. As we discussed earlier, the process of expression of genetic information involves two steps. First step is the transfer of genetic information from DNA to RNA. This process of transfer of genetic information from DNA to RNA is known as transcription. Second step is known as translation. In the process of translation, genetic information is transferred from RNA to protein. The process of translation requires a macromolecular machine known as ribosome. In addition to ribosome, many other macromolecules also participate in the process of translation. First, we shall discuss about transcription. Transcription is a biological process in which only one strand of a double-stranded DNA molecule is used as a template for the synthesis of a complementary strand of RNA. In the process of transcription, only one strand is used as a template strand. The template strand is also known as non-coding strand or antisense strand, whereas other strand is known as coding strand or sense strand. In the second step, that is known as translation, the genetic information is transferred from RNA to protein. The transfer of genetic information from RNA to protein is governed by genetic code. The genetic code is a specification of amino acids by nucleotide triplets known as codons. In the genetic code, totally there are 64 codons. Out of 64 codons, 61 codons specify various amino acids. Some of the amino acids are specified by more than one codon. Remaining 3 codons are called as termination codons. The termination codons causes the termination of protein synthesis. Ribosomes are the macromolecular machines. On these macromolecular machines, the process of protein synthesis takes place. Ribosomes are nucleoprotein organelle composed of 3 to 5 different RNA molecules and 50 to 90 different proteins. The prokaryotic ribosomes are also called as 70S ribosome, whereas the eukaryotic ribosomes are known as 80S ribosome. Both prokaryotic and eukaryotic ribosomes are composed of two unequal subunits, a smaller subunit and a larger subunit. The prokaryotic ribosomes are composed of a larger subunit known as 50S and the smaller subunit is known as 30S, whereas the eukaryotic ribosomes composed of a larger subunit, that is 60S, and the smaller subunit is 40S. This is about ribosome. Ribosome is the organelle that can read the genetic information. There are 5 different classes of RNA. These 5 different classes of RNA are messenger RNA, ribosomal RNA, transfer RNA, small nuclear RNA and microRNA. These 5 different classes of RNA are essential in the process of gene expression. What is mRNA? The RNA that are translated on ribosome are known as messenger RNA. As the name indicates, the messenger RNA carry the genetic information from DNA to the ribosome where protein synthesis occurs. In prokaryote and eukaryote, the process of transcription is similar. The overall process is similar, but the process of transcription in eukaryote is relatively complex. In prokaryotes, the product of transcription is equivalent to mRNA, whereas in eukaryote, the product of transcription is known as primary transcript. These primary transcripts are the precursor of mRNA. So these primary transcripts are also known as pre-mRNA. The pre-mRNA undergo processing. During processing, specific sequences are removed and both the termini undergo modification with the help of various enzymes. Second point is, most of the nuclear gene in higher eukaryotes and some in lower eukaryotes contain non-coding sequences that separate the coding sequences. The non-coding sequences are known as introns and the coding sequences are known as exons. Because in these genes, the coding sequences are separated by non-coding sequences, so this type of genes are called as split genes. Split genes mean a gene that contains both coding sequence and non-coding sequence. So, during the process of transcription in eukaryotes, the entire sequence of split gene is transcribed to pre-mRNA. After transcription, the pre-mRNA undergo processing. During processing, the non-coding sequences are removed by splicing mechanism. And the splicing occurs on a macromolecular structure known as spliceosome. So in eukaryotes, the pre-mRNA undergo processing and modification of both the termini to form mRNA, which can be used for the process of protein synthesis. Friends, this is an introductory account related to transcription and translation. Thank you friends. Please subscribe my YouTube channel Sam Online Class. Thank you very much. See you again. Bye bye.